So, as of right now, researchers have to do a lot of hemming and hawing from different sources to retrieve data and import them to the appropriate programs for parsing. There is also a lot of intuition and hand calculations that have to be done manually in order for them to get a rudimentary understanding of their target genome. There are no easy to use free and or non-programmer friendly tools for generating digitized phylogenetic trees, and there's no visualizations that makes interpreting data equally a cumbersome task. This being the case, the frequency at which researchers can analyze genomes is marginally slower when compared to the computational power of a computer. So, all in all, this program is mostly for genetic researchers, mostly at the University of Washington academia, that helps analyze genomic strands from the GenBank database and cross-analyze different species to create phylogenetic trees and look at where certain mutations happen, where each strand deviates from, parent strand or whatever whatnot. It just simplifies it, makes it a more intuitive GUI, gives you visualizations for better understanding. So the use cases, typically there's multiple different users for this program. Some of them are undergraduate students, some of them are graduate students. A lot of them, all of them are researchers for the most part. I would say that when researchers, especially experienced ones, don't really want to do all the hand calculations and just want to get a quick insight into one of the genomes, using this tool will be very beneficial. So going off with demo, we have the particles.js Genesis splash page. The project name is called Genesis. So essentially, you click on it, and you click sequence, enters. Then you are transported to the first page where you basically choose your reference strand genome, what you want to compare the other strands to. Then you select the comparison strands that you want to compare it to the reference strand. And also, if you look at the bottom, it tells you what kind of sequence analysis is being used and whatever or whatnot to create this analysis. So after you do the 4 drop downs or whatever and you do the sequence, you don't need to select 4, but after you are done selecting the inputs, then you press submit, and then it does all the calculations for you. So if you see at the top strand, there's your reference strand, the one you're comparing the other strands to, and you have the 4 different strands at the bottom, and it tells you where exactly the mutations are occurring, where it differentiates from the parent strand, and also gives you color coordination. So at the top, there's the different types of mutations depending on the color, and also if you put your mouse over some of the letters of 3 amino acid code, it tells you, or the triplet codon. When you put your mouse over the triplet codon, it tells you the amino acids that it's associated with. So this is particularly helpful because there's a lot of associations that you might not be able to do. So then you go into modern evolutionary synthesis, and you're given a basically an equation that you don't have to hand calculate. All you need is the coefficients, and if you look at the bottom, it tells you what's it for mutant gene selection, and it gives you what the coefficients are. So once you actually input, your coefficients that you found from your research, and you press calculate, it gives you a p-value and tells you whether or not the evolution is happening by chance or not. So this is a very rudimentary calculation that's kind of tedious, and maybe researchers don't remember the formula, etc. whatnot. So if you just plug and chug, it gives you the calculations really easy. Okay, so the next part about this is the phylogenetic tree. So once you've gone through all that entire process, it generates a phylogenetic tree based on the parent and the reference strands, and it also gives the phylogen tree, what's it based off of, how we calculate it, what kind of methodology we use, what to expect. We use the UPGMA tree, etc. And then we go on to the frequency chart at the bottom. Well, it gives you synonymous mutations and non-synonymous mutations and tells you, what the definition of each is, and then it gives you like a frequency table. So it tells you what the frequency table of each amino acid is, and when you actually select one of the branches, it actually changes the frequency. It tells you what kind of amino acids compose this genome strand. So this is Genesis. This is the genomic sequencing process, and it's for helping researchers better solve